Hello students, this is Dr. Faizan Mirza. I'm discussing with you the solved questions regarding October-November 2021 variant 12 of your MCQ paper. I'll be discussing question 1 till question 20 in this video. The second half of the question, which is question 21 till 40, will be discussed in the other video. So let's start. So let's start with the paper. This is variant 12 of your MCQ paper. Again, keeping in mind that first 20 questions, normally they come from the first half of your syllabus, that is the first 6 chapters, 5 or 6 chapters, and then the next 20 questions, which are question 21 till 40, they come from the later half of your syllabus. Normally, question these include your chapter 7 till chapter 11. So let's start with the first question now. Now in question 1, they are saying that you have this scale bar and electromicrograph, which is 2 centimeter long. If it's 2 centimeter long, it may mean this is the image given to you, and it represents an actual length of 1 micrometer. So actual measurement is given as 1 micrometer. So how do you calculate the magnification? Magnification is equal to image upon actual. The value here should come as a numerator. This value should go as a denominator. Now, since it is centimeter, you need to convert it to micrometer. How do you convert a centimeter to micrometer? You multiply it by 10,000. So both units in the numerator and the denominator respectively are micrometers. So micrometer gets cancelled, and you simplify this to a value which is about 20,000 magnified. So answer is C. So coming to question 2, eyepiece microscope is fitted with an eyepiece graticle and a straight micrometer scale is placed in the microscope. We know the question somehow deals with the with drawings and stuff, taking measurements. So which statement about stage micrometer, which statements about stage micrometer are correct? So we know stage micrometer is a glass slide. And we place it on the stage. So stage micrometer and slide specimen both cannot be viewed together at the same time. We know the fact. So the scale can be used to measure actual length of the cell directly. No, it cannot be used directly because the eyepiece radical is calibrated using the micrometer, and then you measure the cells with it. You measure the cell with the graticule, which already has been calibrated with the stage micrometer. You can't use the stage micrometer directly for measuring cells. So this is wrong. The scale allows you to calibrate eyepiece graticule. Yes, the scale is visible as objective changes from 10 to 40. Yes, if you increase magnification, so less scale is visible because the scale is as a slide on the glass on the stage. So if you keep on magnifying the field of view, it just keeps broadening, and you can see that the distance which the two points will be apart will keep on moving more distant, so less part of the slide will be visible. You are just magnifying. This is something that can be applied for any slide that you place on the stage, even if it's not scale. If you talk about a specimen, so if you talk about a specimen, less of the specimen will be visible as the objective changes from 10 to 40, and from 40 to, for example, 100, whatever. So it will just keep on increasing. If the magnification is increasing, less and less of that will be visible. So 2 and 3 are correct. 4 students were asked to match the function of the appearance of some cell structure in an animal cell. The function were listed by number. So these are functions, and the appearances are listed by letters. So mRNA passes through the ribosome. mRNA passes through nucleopore. We know we look for nucleopore. Nucleopore is mentioned as X in this list. So double membrane spore interspersed pores. So we know one should be X. So if one is X, that means we look for X here. So the answer could be either C or D. The second option, the second is organized microtubule from spindle. We know the microtubules are organized by a microtubule organizing center or centriole. We look for centriole. So Y here is centriole, non-membrane bound cylindrical structures. So we look for 2 and we look for Y. So D is the option. Again, to confirm this thing, we go for synthesis of polypeptide, they occur on ribosomes. We look for which letter represents ribosome. So ribosome is actually non-membrane bound spherical structures. So we go for 3 as W. So our answer becomes D. Which structures are required for formation of lysosomes? We know lysosomes are specialized Golgi vesicles, and if they are hydrolytic, they carry hydrolytic enzymes. We know that enzymes are proteins, and obviously protein synthesis and the movement will require ATP. So mitochondrial energy is required. Rough endoplasm is required because it will carry out translation of mRNA. Packaging of vesicle will be required because that occurs in Golgi, since lysosomes are specialized Golgi vesicles. So the answer is B. Which cell structures contain ribosomal RNA? Chloroplast, mitochondria, nucleus, they all have ribosomes. Chloroplast and mitochondria having 70 ribosomes each, whereas nucleus having your nucleus is having the ribosomal component of 80 ribosome as well as the subunits there, because the nucleus is having nucleolus. The nucleolus synthesize ribosomes. So you need to just recall that nucleolus is the part of your cell which actually is producing your um, ribosomes. So you can just mention this here that nucleus will synthesize your ribosomes. So nucleus will be having it. So 1, 2, 3 will be the answer. The next question becomes there's a single cell organism. The name is given to you. It measures about 700 micrometer, fine in the diameter, and is enclosed in a cell wall. Okay, the composition of cell wall is not mentioned, so we cannot gauge any information regarding the cell wall. So we move on. The genetic material of this is located free in the cytoplasm. Now that's a major hint that the nucleic acid is in the cytoplasm. There is no nucleus. If there is no nucleus, then the cell must be a prokaryote. Where it occurs in thousands of copies of circular DNA, again, circular DNA is a prokaryote as well. What statement correct? It's a eukaryote. No, it's a plant. It's a plant. All are statements regarding eukaryotes. So all three statements are wrong. It must be a prokaryote. Yes, the option is correct. Ashwin carried out 4 tests on sample of biological molecules. We know that Benedict test for Benedict, we just need to make sure that we go for the basic fact. The Benedict test, you know, it's a test for reducing sugars and for glucose. Glucose is just an example of a reducing sugar. There are many other reducing sugars as well. So the test is not glucose sensitive. So if a solution changes color using Benedict solution, that doesn't mean that that won't mean actually that glucose is present, because any other reducing sugar might be present as well other than glucose. So if emulsion test is giving cloudy, then fat is present. Benedict gives yellows, yes, reducing sugar is present. It doesn't mean glucose is present. Violet purple, protein present. Iodine yellow, starch is absent. What was present? Yes, fat was present. Glucose was present. Not confirmed. We cannot be sure of this. Protein was present. Starch was not present. Yes, the answer becomes 1, 3, and 4. What's the general formula of monosaccharides? That's a fact. It's CH2O and CH2O taken n times. Which statement can be used to describe the structure of cellulose? Cellulose, you know, it's beta glucose flipping hydrogen bond, one glycosidic bond. Just recall all these. It's a polymer of glucose monomers linked by beta 1 4 glycosidic. Yes, it's a polysaccharide of hexose sugar. Yes, hexose means 6 carbon. An unbranched molecule. Yes, it's an unbranched molecule of beta glucose monomer. So all three statements are correct. You can see question 1 till even to question 10. There is no such very difficult question here. So you can go through these questions at a much faster pace. So you have more time for the questions that might be difficult later on in the paper. Complete hydrolysis of polysaccharide require all glycosides to be bonds to be broken. Enzyme X only break 1,4-glycosidic bond alpha, which row shows completely enzyme X can hydrolyze molecule of glycogen and amylose. We know glycogen is a branch molecule having alpha 1 for alpha 1 6 bond. So this enzyme X can break alpha 1,4, but it cannot break alpha 1 6. Amylose, on the other hand, we know it's helical and it's having 1,4 bond only. So amylose will be broken fully by the enzyme X, where glycogen will be broken partially. Which row will describe this? We know glycogen will give some hydrolysis, whereas amylose will give most hydrolysis. So A is the answer. Which molecules contain at least 2 double bonds? At least 2 double bonds are present in protein, because the proteins are having C double bond O and N and H. So wherever this C double bond O keeps suffering, so that means that we'll be having 2 double bonds. So saturated fatty acids, collagen, hemoglobin, yes, but not saturated fatty acid. Collagen, yes. Saturated fatty acid, no. Only no. Hemoglobin, collagen, yes. Saturated fatty acid again, so yes, hemoglobin and collagen are the answer here. Again, there's an answer. There's a question for proteins. So there are 2 questions for proteins here. This one is actually for amino acid. So the diagram here shows an amino acid. Which group is changed to produce a different amino acid? You have to identify the R group. We identify the R group in such a way that there's in an amino acid there's always a center carbon. The center carbon is identified with the C double bond OH, which is the carboxyl end of your amino acid. This is the carboxyl end of the amino acid. The same carbon will have amine end at the opposite end. So that's the amine end. So we know that, okay, this is our central carbon, and we need to look at this carbon, and this carbon will have the third bond with the hydrogen. All these structures will always be the same in the amino acid. The only differing structure will be at the fourth bond, which is the R group. So this is, in this case, methyl group, or CH3 is the group, which is the R group. So if you change the R group, amino acid will change as well. Which row is correct about structure of proteins? We'll just read through all these statements. Primary structure, we know it's the order of amino acids. The sequence, yes, divided by nucleotide. This is correct. Secondary structure occurs by hydrogen bonding between NH and CO groups of amino acids. This is perfectly right as well. Disulfide ionic hydrogen bond hydrophobic interactions, they all are involved. Yes, in tertiary structure. Quaternary always formed by association of 2 or more polypeptides. All 4 statements are correct. Now, if you're not very sure, let's go for looking for why B, C, and D are wrong. B says that primary structure is the number of amino acid. No, it's the order. In a single poly, so if it's wrong, this we won't go for any further because we know that's wrong. In C, The first is fine. Yes, the amino acids are linked by a specific order. They say an alpha helix is formed by hydrogen bond. Yes, this is good for secondary structure. But then they are saying between the side chains of the amino acid, which means between the R groups. No, secondary structure is never because of the R groups. Secondary structure is only because of the carboxyl and the amine groups present in the peptide bond of amino acid which are lying close together. So C is wrong as well. In D, again, the first option is correct. Again, the second option in the second structure is wrong. It's a beta sheet. Okay. Resulting in folding. No, it won't fold. It's just folded partially. It's not a complete three-dimensional folding. Forming hydrogen bond between adjacent strand. Okay. Even if we take that, okay, fine, this folding is correct. There is no concept of multiple strands being there together in a secondary structure. Primary, secondary, tertiary structures are always for a single polypeptide. It's always single polypeptide. And if there are more than one polypeptide, every polypeptide will have primary, secondary, tertiary together. And for every polypeptide having primary, secondary, tertiary together, they will all combine together to form the quaternary structure. So it's always quaternary is about formation of a protein with multiple polypeptides, each individual polypeptide having its own primary, secondary, and tertiary folding. Question 14. Which graph correctly show possible changes in energy level of a chemical reaction progress with or without enzyme? Now we know with enzyme the peak should be lower. Without enzyme the peak should be higher. So in this case they have normally what do? We use the dotted line for with enzyme and a continuous line for without enzyme. So first trick the examiner is saying is this. So they have changed the key. So make sure that you get the key. You mentioned lower peak and higher peak here. So going from here, you just go to A and dy. A and D are wrong because this reaction, the energy level cannot differ. This is not possible that the products can have different energy level with and without enzyme. So A and D are outright wrong. What's wrong with B? In B, you can see they are saying that this continuous line, which is with enzyme, it's having a higher peak. That's not possible because we know enzymes lower the activation energy. So when they lower the activation energy, yes, C is correct because you lower peak with enzyme, there's a higher peak without enzyme, and the graph goes to the same point for both enzyme and without enzyme. The graph shows a relationship between concentration of a substrate and the rate of enzyme catalyzation. So you are increasing the substrate concentration, the rate of reaction increases and eventually becomes constant and parallel to the x-axis, which shows Km and Vmx of this enzyme would be affected if the same reaction was carried out in the presence of competitive inhibitor. We know the enzyme will have the same Vmax. It will have an increased Km, which means there is a lower affinity. So the graph will be something like this. It will go to the same Vmax. Vmax is at this point. It will still have the same Vmax. We calculate the Vmax from this point on the y-axis, and half of that will be our half Vmax, and from that we actually come to our Km. This is again described in various lectures of mine in previously as well. We have covered in enzymes as well. So we know Km increases and Vmax remains same for competitive inhibitors. This is fact and use scientific fact. So yeah, question 16. Liver cells contain vesicles. If liver cell contain vesicles, so for example, if this is a liver cell, this is having vesicles. Okay. The vesicles that have proteins in their membrane, which are specific for transport of glucose. So these are vesicles present inside the cells of liver, and each vesicle is having transport proteins. These are transport proteins present on the vesicles present inside the cytoplasm of the liver cell. This is the description already given to you. Now, coming back. The question further states that when these cells need to take up glucose, the vesicles fuse with the cell membrane. How does the uptake of glucose occur? We know glucose is a polar molecule. If it's a polar molecule, can it enter by exocytosis? No. Can it enter by diffusion? No. Can it enter by endocytosis? No. It will occur. It will go by passive diffusion. Why? Because this vesicle will start to move to the cell membrane. It will attach to the cell membrane and the channel, the glucose transport protein. This is the glucose transport protein. Glucose transport protein. This glucose transport protein will become the part of the cell membrane, and then glucose will start to enter from a high concentration which is outside to a low concentration which is inside. So this is how the glucose will start to enter through this protein channels. So this question actually confuses the student by giving the option of exocytosis, because normally the students of AS, they know that the vesicle, if it's in the cytoplasm, it will go to the membrane, it will release contents outside by exocytosis. Keep in mind, there is no exocytosis occurring. the vesicle is going to the membrane, becoming a part of it. It is not releasing anything outside. Since it's not releasing anything outside, exocytosis cannot be the answer. Question 17. Now, question 17 would need some working here, and I have not included all the working though. I am just describing to you how this is happening, so you can just probably do the working on your own. A student set up an experiment to investigate diffusion of block of agar 1 by 1 cm. CM was stained uniformly with water soluble blue dye. So there is a cube, and this cube is having these dimensions, and it's having a blue dye. The block of agar was put in a test tube containing 10 cm of distilled water, which is, we know distilled water, so it's clear. So we allow at 10-20 degrees C, the intensity of blue color of water after 5 minutes was measured. So we know that if you have a clear distilled water and this blue cube is placed in it, the blue cube would lose this blue dye to the distilled water, and gradually the blue color will be there in water after 5 minutes. We can measure its intensity after 5 minutes. So we have this block and we have blue solution. Four other experiments A, B, C, and D were carried out using different number of agar blocks, different sizes of agar blocks. So there you are increasing the number, you are changing the size, and at different temperature. There are three variables you are changing. You are changing the number, you are changing the size, you are changing the temperature. All of the variables were standardized. Now we need to recall how diffusion is affected by all of these. So, which experiment would have a lighter blue color in water after 5 minutes? Lighter blue color means that the block is retaining the blue dye, the block is not giving off the blue color very readily, and the solution is not turning the same intensity of blue in 5 minutes. So, that means that the block must have a larger surface area to volume ratio. The temperature must be low. There must be more than one blocks present, because that would decrease the gradient at which the exchanges are going. For example, I think the largest surface area and temperature being low are something that comes naturally to you. We know if largest surface area to volume ratio is there, the exchange will be slower. At lower temperature, there is lesser kinetic energy, so again exchange will be lower. This is something that can trick the students, that more than one blocks. How is this affecting the rate of diffusion? You can take an example of sugar crystals. For example, if you have one glass of water and you try to dissolve just one teaspoon of sugar crystals in it, you can dissolve them. But if you add 2 teaspoons of sugar crystals, or 3 teaspoons of sugar crystals, that means you are increasing the number of crystals of sugar in that limited amount of water. Now, initially, the concentration gradient will be very steep, but then the sugar crystals will just not dissolve. They will not dissolve because the solution will become saturated. The gradient will decrease. The gradient will not be as steep as it was for 1 teaspoon sugar in one glass of water. So the same thing goes here. So if we go for the size of blocks and the temperature and the number of blocks, so temperature 30 degrees Celsius cannot be the answer, because 30 will provide a higher kinetic energy. Diffusion will be much faster. The answer must be for 20, which will be slower diffusion. Now you have to pick between B and D. Now we know there are two blocks, agar blocks in B, and there's one here. The answer should be 2, because the two blocks will reduce the concentration gradient. Now, still, if you want to go for a working, then you need to recall what the formulas are for calculating surface area to volume ratio. If you are to calculate the surface area to volume ratio of a cube, you use a formula of 6a square upon a cube. For instance, I had 1 cm cube 1 by 1 by 1 cube here. So my working would be 1 squared upon 1 cube. And when I do this, my answer turns out to be 6. When I do the same for this 0.5 by 0.5, my answer comes out 12. So this is a larger surface to volume ratio, and this is exactly what we are looking for. Now, if you think that D is having a larger surface area as well, you can apply not this formula, because this formula is for cube. You will apply the formula for cuboid. Cuboid formula is 2 into length into breadth plus breadth into height plus length into height divided by length into breadth into height. What's a cuboid? Cuboid is a rectangular three-dimensional structure. So this is a cuboid. So C and D are cuboids, whereas A and B are cubes. You can just perform this calculation and see how this goes on, and that actually will tell you that this will be carrying out diffusion faster as compared to B. So B will be our answer. Which description is correct? A centromere hold two chromatids together until the end of prophase. No, it holds the chromosome until the end of metaphase. A is wrong. A chromatid is one of the two identical part of chromosomes and is made up of proteins. Yes. And two DNA molecules. No. A chromatid is made up of a DNA molecule. Number of chromatid is always equal to number of DNA molecule. You can just make this as a universal fact. A chromosome is a structure with two identical parts made of DNA and proteins found in the nucleus of a prokaryotic cell. This is just not scientific. Prokaryotes do not have nucleus. A telomere is a sequence of DNA nucleotides such as GGG, okay, yes, that's very well possible. Repeated many times, yes, and found at the end of each chromatid, perfect. So D is the answer. During metaphase, the scientists change the chromosome of a diploid animal cell with a fluorescent dye to allow the telomeres to be observed. The cell is having 26 chromosomes. How many telomeres will the scientist observe? How a metaphase chromosome looks like? How an anaphase telophase chromosome looks like? You need to make sure that you remember these diagrams. An anaphase and telophase chromosome looks like something like this. There is a centromere. The chromosome is having a single chromatid with two telomeres, one at each end. Whereas when we go for a metaphase chromosome is having a centromere and there are two chromatids, and each chromatid is having one telomere at one end. That makes a total of 4 telomeres at one chromosome. So if there are 26 chromosomes in metaphase, we'll multiply 26 by 4, we get the answer 104. The photomicrograph shows cells of various stages of life cycle of a cell. You just identify these. This is metaphase. The chromosome can be seen lined up along the center. Nuclear envelope is just not there, so that must be metaphase. Late anaphase, why? Because the chromosome have been pulled apart. Yes, they have been pulled apart. So this is late anaphase, or probably this is actually not just late anaphase. This is telophase. We can identify this as telophase as well, because there is, you can see this very fine nuclear envelope forming if you look closely. So yes, telophase is there. Okay, a prophase is there, and anaphase is there as well. Which cell contain twice as many DNA molecules as the cell or the organism that has just finished mitotic cell cycle ending with cytokinesis? Now, since the cells have not separated, answer will be 1, 2, and 3, and 4, all of these. That's it from my side for this video. That's it. Thank you so much.